Hello, welcome to BioTanime. Measurement of antigen-antibody complex. Different serological reactions are precipitation reactions, agglutination reactions, precipitation reactions. Different types of precipitation test are ring test, slide test, tube test. Ring test is performed in a test tube where the antibody solution to be detected in the sample is put at first and antigen solution is poured. The test is confirmed as positive by the observation of a precipitation ring in the middle of the tube after a few hours. The ring test is used in the Lancefield technique, for grouping Streptococcus species. Also used in the detection of anthrax by Ascoli's test. Slide test. This test is performed on slides. In this test, the serum sample of the suspected patient is kept in the cavity of the slide and already known antigenic solution is mixed with it and shaken properly. In the case of test, floccules are formed after a while. It is mostly used for detection of syphilis antigen in suspected patient. The test is called the VDRL test or Venereal Disease Research Laboratory test. Tube test. Type of flocculation test in which antigen or antibodies solution are mixed in a tube to observe the formation of floccules. Example: Kahn test, used for the detection of syphilis. Precipitation reaction in agar. It is performed on agar or agarose gel. It also termed as immunodiffusion. Gels provide a medium for the diffusion of reactants through the pores. It is 4 types. 1. Auden method. 2. Radial immunodiffusion. 3. Oakley-Fulthrope technique. 4. Autaloni technique. 1. Auden technique, also known as single diffusion in single dimension. In this type, only one of the reactants is allowed to diffuse in a single direction towards their respective antibody. One of the reactants, that is, antibody, is mixed with the gel medium and kept in a test tube with a temperature of around 60 degrees Celsius. After cooling it for a while, the antigen solution is kept. The antigen diffuse below and form a precipitate ring with antibody. 2. Radial immunodiffusion. Single diffusion in double direction. In this, agarose gel is mixed with antibody solution. It is then poured on a Petri plate. Well is made in the medium and antigen solution is poured into the well. 3. Oakley-Fulthrope technique. Double diffusion in a single dimension. In this technique, the antibody is at first mixed with agar and kept in the test tube. Another layer of plain agar is laid over it and antigen solution is then poured over it. The antigens and antibody both move towards the plain agar layer between them to interact. 4. Autaloni technique. Double diffusion in dimension. In this technique, the gel is at first poured on a Petri plate and one well is made at the center, and around the central well, multiple wells are made. In the center, antibody solution is poured and antigen solutions are poured in other wells. It is then kept for incubation for 1 or 2 days in a moist chamber. The antigens and antibodies both diffuse towards each other in 2 directions, that is horizontal and vertical. Precipitation reaction in agar in electric field. Involves the precipitation in agar but with an addition of a current or electric field. Different reactions include in this technique are 1. Immunoelectrophoresis. 2. Counter-current electrophoresis. 3. Rocket electrophoresis. Precipitation reaction applications. Used in diagnostic immunology, detection of syphilis in patients by VDRL test, Kahn test, etc. In the separation of specific proteins by precipitating them using their specific antibodies. In the grouping of different microbes such as Streptococcus based on the presence of different antigens. Standardization of the toxins with their respective antitoxins. Agglutination reactions is the visible expression of the aggregation of antigens and antibodies. Various methods of agglutination are used in diagnostic immunology. These include 1. Latex agglutination. Many antibody molecules are bound to latex beads or particles, which increases the number of antigen binding sites. If an antigen is present in a test specimen, it will bind to the antibody and form visible, cross-linked aggregates. 2. Direct bacterial agglutination uses whole pathogens as a source of antigen. It measures the antibody level produced by a host infected with that pathogens. The binding of antibodies to surface antigens on the bacteria results in visible clumps. 3. Heme agglutination uses erythrocytes as the biological carriers of bacterial antigens, and purified polysaccharides or proteins for determining the presence of corresponding antibodies in a specimen. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.